Hello everyone, myself Dr. Deepika Saini, Associate Professor at Lloyd Institute of Management and Technology, Greater Noida. I am going to present a lecture on antipsychotic agent. One of the subtopic under the unit 4 of Medicinal Chemistry 1 course code BP402T. In the previous lecture, we have completed hypnotic and sedative. So, first of all, what are antipsychotic drugs? When we talk about antipsychotic drugs, these are the drugs which are used to treat psychosis. These are also known as neuroleptics and major tranquilizers. But basically, antipsychotic drugs which are used to treat schizophrenia in patients. Unfortunately, schizophrenia is for life. Like, we have not yet developed any drug which can treat schizophrenia. We can only decrease the intensity as well as the extent of schizophrenia in the patients. We can decrease the frequency of manic attacks in the patient, but we cannot treat the patient completely. These antipsychotic drugs are also effective in other psychotic states like mania, paranoia, hallucinations and delusions. So, in these states also we can use antipsychotic drugs to produce the calmness in the patient, to reduce the intensity of hallucination as well as delusions. Hallucinations is a state when a patient start imagining a different world. In the case of delusions, they start imagining the wrong hypothesis that is not even exist in reality. They start thinking, for example, they will start thinking about an incident that actually did not happen. So, in these of the cases, patient with schizophrenia, they are not able to perform well in society. So, by the use of antipsychotic drug, at least that will support the patient to work as a normal human being in the society. Mechanism of action of antipsychotic drug. The basic mechanism of antipsychotic drug is dopamine antagonism. Basically, they antagonize the action of dopamine. All the antipsychotic drugs except clozapine, all the drugs will have a blocking action on D2 dopamine D2 receptor in the brain as well as in the periphery. They will block the action of dopamine. For example, dopamine, when it releases in synaptic cleft, antipsychotic drug will replace them and will itself join at the dopamine receptor and will block its binding as well as its activity at the receptor and hence decrease its response. Dopamine is responsible for any activity, we would say the hyperactivity in the body. So, when any drug that will block the dopamine action will definitely induce the calmness in the patient or the recipient. Further, these drugs has been classified into different category of drug, like first of all phenothiazine derivatives, next is fluorobutyrophenone derivative, next is beta aminoketones, benzamides and the miscellaneous agents. First of all, the category phenothiazine, that has an example of promazine, chlorpromazine, clozapine. Clozapine does not act on dopamine receptor, it acts through histaminic receptor, but also possess the same antipsychotic activity. Fluorobutyrophenones example is haloperidol, one of the most common drug to be used as antipsychotic. Beta aminoketones, molindone, hydrochloride, benzamide derivative is sulpiride, and miscellaneous agents like olanzapine and oxypertin. The first category is phenothiazine. First of all, we will try to understand its structure. It's phenothiazine. Pheno means it contains phenyl rings. Thia means it contains sulfur. Pheno means phenyl rings, and it's aza. Aza means nitrogen. So, it contains phenyl ring as well as sulfur as well as nitrogen. So, in this structure we have clearly seen two benzene rings had sandwiched a sulfur and a nitrogen atom. So, here it is a phenyl ring, it is a phenyl ring, and in between we have sulfur and nitrogen. When we start this number from here, 1, 2, 3, 4, 5, again we have left, like in the last lecture I have told you, when we number a fused system, we do not go for a numbering at the fusion phase. So, it starts 1, 2, 3, 4, then 5, 6, 7, 8, 9 and 10. The 10th position is for nitrogen. So, we have given the number accordingly, the second position for the substituent, and our 10 position means nitrogen is being substituted. So, in case of phenothiazine, first example is bromazine, in which this carbon atom is unsubstituted and this nitrogen atom is substituted with 2 methyl rings. Further, the chlorpromazine, the structure is exactly similar to the promazine. The only difference is it contains a chlorine atom. So, again there is 2 methyl substitution at the nitrogen. Further, triflupromazine, it contains it contains 3 fluorine atoms substituted with a carbon at position number 2, and again the nitrogen is being substituted with 2 methyl groups. These are further examples of phenothiazine. This is the example of thioridazine. This is piperacetazine, in which this nitrogen is further substituted with a piperazine ring, and this is prochlorperazine. This is one of the common drug that is being used in the treatment of psychosis. So, now we will move to the structure activity relationship of phenothiazine. When we talk about the structure activity relationship of phenothiazine, we have 3 different targets. First of all, any modification in a phenothiazine ring, that if we can modify this phenothiazine ring. Second, modification in the alkyl side chain, this alkyl side chain. Further, the amino group modification. Further, when we modify the amino group. So, first we will consider the nature of alkyl side chain. When we talk about nature of alkyl side chain, potency is maximum when we differentiate this nitrogen with this nitrogen at least with the 3 carbon atoms. If there are 3 carbon atom gap between the 2 nitrogen atoms, that compound will possess the maximum potency. By the introduction of any methyl group at carbon number 1, this carbon number 1 that will decrease the antipsychotic potential of the drug, and that will produce imipramine like activity, means that may have anti-depressant action. So, apart from that, when even the oxygen atom is introduced into the carbon atom number 1, that will also result in potent antidepressant action. Rather depressing the central nervous system, they will start acting as antidepressant effect. Example is chloracetine is example of such drug. Further, if we add any methyl group at position number carbon number 2 or carbon number 3, that will not bother any kind of activity at this position. So, again repeating, we have 3 different positions, first, second and third. First of all, the first point to be keep in mind that the potency is maximum when there is difference of 3 carbon atom between the 2 nitrogen in the phenothiazine derivative. Next, if we talk about the substitution on these carbon atoms, when we introduce any methyl group or any cyclopropyl ring at position number carbon number 1, that will decrease the antipsychotic activity, and further it produce imipramine like effect, that is not going to be antipsychotic, that will produce antidepressant effect. Even if we introduce oxygen at position number 1, again it produces antidepressant effect rather than antipsychotic. And if we do any changes at position number 2 or position number 3, that will not affect the antipsychotic effect of phenothiazine. Further, the amino group of side chain. When we talk about the amino group, the tertiary nitrogen atom that shows maximum potency rather than secondary and primary. If we have all the substitution at position number this nitrogen, if we substituted this nitrogen with both the R1 and R2 are substituted, means this nitrogen is a tertiary nitrogen that possess maximum activity. But if we have any unsubstituted, like if we have hydrogen in place of either R1 or R2, either it is a primary NH2 or it is NHR secondary, that will lead to reduced or sometime abolished activity. Activity is also decreased when a dimethyl amino group is replaced by any morpholine group or the thiomorpholine group. In these cases, activity will get reduced. However, if we use piperidine or piperazine, that will lead to increase and will have more potent compound, even much better than the NCH3 twice group. So, in case we want to increase the potency of a compound, we should replace this nitrogen NCH3 twice group with either the piperazine or with the piperidine ring. And these bridged piperidine derivatives that retain high degree of activity, even the introduction of OH group, CH3 group, methyl group at position number 4 of piperazine, that will also lead to increase in activity. So, at this position, this nitrogen with 2 CH3 group, they will have lesser activity, lesser potency as compared to with a nitrogen atom with a piperazine ring. And if we replace, we also substitute this nitrogen either with OH or CH3 or anything else, that will also increase the activity. Now, when we talk about the first one, the substituent on the aromatic ring. So, any substituent at position number 2, this position number 2, that is optimum for the neuroleptic activity. Neuroleptic activity means antipsychotic activity. So, when we consider the the substitution at various positions, we have seen the substituent at position number 1 have the least active compounds as compared to 4 position, as compared to 3, and the position number 2, this position has the maximum potency when we substitute the compounds, the substituent at position number 2. Further, the potency of various group increase in a following order as shown, like OH has least activity as compared to hydrogen, as compared to cyanide, as compared to methyl. Further, it get increased with respect to chlorine, and when we have substituted with triflu, that is CF3, that has possessed maximum activity. Further, dye substitution of carbon number 2 substituent drug that lead to toxicity. These are the drugs developed after the dye substitution that has harmful potency, harmful effect rather than antipsychotic activity. So, when we look back, we have 3 options. One is nature of alkyl chain, we have next is amino group substitution and the aromatic substitution. First of all, in the case of nature of alkyl side chain, we must keep 2 nitrogens away by 3 carbon atoms, and in these carbon atoms only position 2 and position 3 can have any methylation that will not possess any effect on the activity, that will not alter the activity, but at position number 1 that will definitely decrease the antipsychotic activity, rather in some cases that has also produced antidepressant effect. Further, in case of amino, as we have discussed this amino side chain, we have seen that the tertiary compounds, when the nitrogen atom is replaced, is tertiary in nature, means the R1 and R1 dash is being replaced by any of the 2 alkyl group at least that possess a better activity as compared to secondary, that is NHR, as compared to primary like NH2, they have shown reduced or in some cases there is no activity at all. Activity is also increased by the replacement of this nitrogen atom by any morpholine group or thiomorpholine group, but we can replace this nitrogen by piperidine or piperazine. The compound with the substitution with the piperidine and piperazine, they found to be more potent, and apart from that the 4th position, like they have been attached from here. So, 1, 2, 3, 4 by substitution of introduction of OH, CH3, ethyl or CH2OH at carbon number at this position, the position number 4, that will also increase in activity. And in case of aromatic ring, substitution at carbon number position number 2, that is very good for the neuroleptic, for the antipsychotic activity, though this sequence, the first position is the substitution at first position sounds to be least active, and substitution at position number 2 will produce more active compound. In case of different substitution, we have seen the order also, like OH will produce the least active compounds as compared to hydrogen, as compared to cyano group, as compared to methyl, as compared to chloro, as compared to CF3 group. These are some ring analogues of phenothiazine. Example is chlorprothiazine and thiothiazine. These are the examples of ring analogues of phenothiazine. Loxazepam and clozapine are the examples, another examples of ring analogues. So, now this is the synthesis of chlorpromazine. First of all, we have taken 3-chlor-diphenylamine. First, we have to draw the structure of diphenylamine. This is diphenylamine. So, this amine group is attached to this will go 1, 2 and 3. We have taken 3-chloro. So, this is chloro. So, 3-chloro-diphenylamine. By the action of sulfur, what will happen? There is this ring get fused by the introduction of sulfur atom. Now, we have 2-chloro-phenothiazine, because in case of phenothiazine we will start 1, 2, 3, 4, 5, 6, 7, 8, 9 and 10. So, it is 2-chlorophenothiazine. When this 2-chlorophenothiazine, that will get reacted with 3-dimethylaminopropyl chloride. First of all, we will prepare CH, I will write 2 though it is 3 because it is substituted. So, it is a propyl chloride. We have and this is 3-dimethylamino. First position, second position and third position, and at third position there is dimethylamino, means at nitrogen there are 2 methyl substitutions. So, this is propyl chloride and this is dimethyl amino group. So, 3-dimethylaminopropyl chloride, this is 3-dimethylaminopropyl group. So, by the action of NaNH2, it will get converted into chlorpromazine. So, simply this hydrogen, the abstraction of this hydrogen and this chlorine minus HCl, and this directly this carbon atom is now attached here. So, it has the structure of chlorpromazine. So, the synthesis of chlorpromazine, in this synthesis 3-chlorodiphenylamine is by the action of sulfur, by heating with sulfur, it get converted into 2-chlorophenothiazine. This 2-chlorophenothiazine is further treated with 3-dimethylaminopropyl chloride, and by the removal of HCl, that will lead to the formation of chlorpromazine. So, this is synthesis of chlorpromazine. Now, have a quick glance at the today's lecture. First of all, antipsychotic drugs, these are the drugs that will act against schizophrenia and that will act also under the effective for the other psychotic states like mania, hallucination, delusions, and unfortunately, as we have discussed, schizophrenia is for life, that we have not yet developed any drug that is can completely cure schizophrenia in patients. We can only decrease the intensity and the frequency of manic attack of the patients and the recipients, and we can decrease the intensity of hallucination and delusions that they have and let them live a supportive life in the society. Mechanism of action, that they are a very well antagonizes the dopamine action. What will happen? They will not allow the dopamine to bind at its receptor, rather they will bind by itself, replacing the dopamine. Hence, dopamine will not possess the excitatory action, and hence the antipsychotic activity, antipsychotic agents possess their activity. Further, the antipsychotic agents, they have been classified into various categories like phenothiazine derivatives, fluorobutyrophenones, beta aminoketones, benzamides, and the miscellaneous agents. The first category under study is phenothiazine. There are several examples of phenothiazine like promazine, chlorpromazine, and triflupromazine. Further, these are the examples of phenothiazine: thyrodazine, piperacetazine, and prochlorpropazine. pyrazine is the drug are the drugs of this category. When we talk about structure activity of phenothiazine, there are three basic sites that we can target to have structure activity relationship. To know the structure activity relationship of phenothiazine, we can change, we can modify the alkyl side chain, we can do alteration in the amino group. Apart from that, we can also modify phenothiazine ring. First, the nature of alkyl side chain that we have studied, that the two nitrogen atoms, these two nitrogen atoms must be away from at least 3 carbon atoms. There must be 3 carbon atoms in between the two nitrogens of phenothiazine derivatives. If we do any alteration in carbon number 1, that will decrease the antipsychotic activity, and in some cases it also produces imipramine like activity, or we can say antidepressant activity rather than CNS depression action. Further, any addition at carbon number 2 and carbon number 3 with the alkyl group or the CH3 group, that will not alter the antipsychotic potential of these drugs. Further, when we talk about the amino group of the side chain, the amino group must be tertiary, the secondary and primary nitrogen atom, that will have a reduced or the abolished activity. Apart from that, when we replace this dimethyl amino group with the pyrolinidine or the morpholinil or the thiomorpholinil group, that will decrease the activity. If we wish to increase the activity to have more potent compound, we must replace this dimethyl amino group with the piperidine or the piperazine derivatives. Further, this bridging of piperidine and piperazine, they have possessed the high degree of activity, and even the introduction of OH, CH3 and other groups at position number 4 of piperazine, that will lead to increased potency of antipsychotic agent. When we talk about the substitution at the aromatic ring, this phenothiazine ring, we have several options, like we can make any changes at these 4 positions. But when we did, we came to know about the order in which the potency can be altered. Any changes at position number 1, that will lead to least active compound rather than 4, rather than 3, and the most potent compound was found with substitution at carbon number 2. And the potency of various group, that is also being studied, and following order was formed, like OH, H, CN, CH3, Cl and CF3 was found to be most active. These are the ring analogues of phenothiazine. So, this is the synthesis of chlorpromazine, in which 3-chloro-diphenylamine is heated with sulfur, will get converted into 2-chlorophenothiazine, and this 2-chlorophenothiazine is further reacted with 3-dimethylamino propyl chloride, and by the removal of HCl in the presence of NaNH2, chlorpromazine is formed. Thank you. In the next class, we will study about the various other classes of antipsychotic agents. Thank you.